Today we will be giving a tour of a common eukaryotic cell. This presentation is narrated by Broach,
Krystal Favourite,
Enyinnaya Kamalu,
Aditya Natu,
and Tarik Prince.
Facilitated diffusion. Facilitated diffusion is also known as facilitated transport or passive mediated transport. It is the process of spontaneous passive transport of molecules or ions across a biological membrane via specific transmembrane integral proteins. Since it's passive, it does not directly require chemical energy from ATP hydrolysis in a transport step itself. The molecules and ions move down, their concentration gradient reflecting its diffusive nature. Peroxisomes. Peroxisomes are similar to lysosomes. They are small vesicles found around the cell. They have a single membrane that contains digestive enzymes for breaking down toxic materials in the cell. They differ, however, from lysosomes in the type of enzyme they hold. Peroxisomes hold on to enzymes that require oxygen, which are oxidative enzymes. Peroxisomes absorb nutrients that the cell has acquired. They are very well known for digesting fatty acids. They also play a part in the way organisms digest alcohol or ethanol. Because they do that job, you would expect liver cells to have more peroxisomes than most other cells in the human body. They also play a role in cholesterol synthesis and the digestion of amino acids. Peroxisomes work in a very specific way. Their enzymes attack complex molecules and break them down into similar molecules. One of the byproducts of the digestion of hydrogen peroxide, peroxisomes have developed to a point where they are able to contain that hydrogen peroxide and break it down in water and oxygen. The water is harmless to the cell, and the oxygen can be used to the next digestive reaction. Peroxisomes have a single membrane that surrounds the digestive enzyme and dangerous byproducts of their work. The protein enzymes are usually created by lysosomes floating in the cell. They then insert the protein into the peroxisome bubble. Peroxisomes continue to grow until they split in two. Where does the membrane come from? Scientists are still researching that answer. It may come from the endoplasmic reticulum, but it may be created in a way different from lysosomes. Mitochondria, endosymbiotic theory. Mitochondria are known as the powerhouse of the cell. They are organelles that act like a digestive system which takes in nutrients, breaks them down, and creates energy-rich molecules for the cell. The biomedical process of the cell are known as cellular respiration. Many of the reactions involved in cellular respiration happen in the mitochondria. Mitochondria are the working organelles that keep the cell full of energy. Mitochondria are small organelles floating free throughout the cell. Some cells have several thousand mitochondria, while others have none. Muscle cells need a lot of energy so that they have loads of mitochondria. Neurons, which are cells that transmit nerve impulses, don't need as many. When a cell feels it is not getting enough energy to survive, more mitochondria can be created. Sometimes the mitochondria can grow larger or combine with other mitochondria. It all depends on the needs of the cell. Mitochondria structure. Cross-section of a mitochondrion, membranes, matrix. Mitochondria are shaped perfectly to maximize their productivity. They are made of two membranes. The outer membrane covers the organelle and contains it like a skin. The inner membrane folds over many times and creates layered structures called cristae. The fluid contained in the mitochondria is called the matrix. The folding of the inner membrane increases the surface area inside the organelle. Since many of the chemical reactions happen on the inner membrane, the increased surface area creates more space for reactions to occur. If you have more space to work, you can get more work done. Similar surface area strategies are used by microvilli in your intestines. But what is the matrix? It's not like the movies at all. Mitochondria are special because they have their own ribosomes and DNA floating in the matrix. There are also structures called granules, which may control concentrations of ions. Cell biologists are still exploring the activity of granules. Mitochondria continued. Exactly how does cellular respiration occur in the mitochondria? The matrix is filled with water and proteins or enzymes. Those proteins take organic molecules such as pyruvate, inositol, CoA enzyme, and chemically digest them. Proteins embedded in the inner membrane and enzymes involved in the citric acid cycle ultimately release water and carbon dioxide molecules from the breakdown of oxygen and glucose. The mitochondria are the only places in the cell where oxygen is reduced and eventually broken down into water. Mitochondria are also involved in controlling the concentration of calcium ions within the cell. They work very closely with the endoplasmic reticulum to limit the amount of calcium in the cytosol. The evolutionary origin of mitochondria. Unlike any other organelle except for chloroplast, mitochondria appear to originate only from other mitochondria. They contain their own DNA, which is circular, as is true with bacteria, along with their own transcriptional and translational machinery. Mitochondrial ribosomes and transfer RNA molecules are similar to those of bacteria, as are components of their membrane. These and related observations led by Dr. Lynn Margulis in the 1970s to propose an extracellular origin of mitochondria.
Active transport is defined as a process in which molecule moves across a cell membrane from a region of low concentration to a region of high concentration. This upward movement of substances against the concentration gradient requires energy and hence is called active transport. During this process, cells inside a membrane move substances through transport proteins. Some of these proteins act as pumps, that is, they use the energy to move substances in and out of the cell and hence maintain the desired concentration gradient. For example, the sodium-potassium pump. Here, within the cells, the sodium is kept in a concentration that is much lower than the exterior environment. This, on the other hand, is opposite for potassium. Hence, sodium and potassium have to move against the concentration gradient. The sodium moves actively out of the cell, whereas the potassium moves actively inside the cell. This takes energy, which comes from the chemical energy stored in adenosine triphosphate, which is also known as ATP. The cytoskeleton. Cytoskeleton is collectively defined as a network of protein fibers which are responsible for maintaining the shape of the cell, secure some organelles in specific position, allow cytoplasm and vesicles to move within the cell, and enable cells within multicellular organisms to move. There are three types of filaments in the cytoskeleton, namely microfilaments, microtubules, and intermediate filaments. All of these combined share important features which, when combined, provide collective productivity to the cytoskeleton. Beginning with the first one, microfilaments. Of all the fibers, these are the narrowest. These are responsible for cellular movements of the cell. They are about 7 nanometer in diameter. Microfilaments provide some rigidity and shape to the cells and also can be disassembled and assembled, which helps the cell to change its shape at its discretion. An example of microfilaments are the white blood cells. They make great use of this cellular dynamics to reach to the site of infection and phagocytosize the pathogen. Second, intermediate filaments. These are made up of collective strands of fibrous proteins. The diameter ranges from 8 to 10 nanometers and lie within the diameters of microtubules and microfilaments. They, however, do not have a role in cellular movements. Intermediate filaments' primary role is to bear the tension and hence maintain cell structure. They also help in anchoring nucleus and other organelles in place. An example of this is keratin, the fibrous protein that strengthens your hair, nails, and epidermis of the skin. Finally, microtubules. These, as the name suggests, are small hollow tubes. The wall constitutes of dimers of alpha-tubulin and beta-tubulin and 2 globular proteins. These are the widest of all and around 25 nanometers in diameter. They help the cell resist compression and provide a track along which vesicles move through the fiber. An example of these are polar fibers. Centrioles. These are defined as cylindrically shaped cellular organelles and are found in most eukaryotic cells. Centrioles are seen during the process of mitosis and meiosis. These are typically located near the nucleus in the centrosome. Centrioles are made of 9 bundles of microtubules that are arranged in a ring. The main function of centriole is to help with the cell division in animals. It can be interesting to learn that the absence of centrioles causes division errors and delays in the mitotic process. It was found that centrioles have evolved as a cell refinement, which makes mitosis a more efficient and lesser error-prone process.
The cell membrane/plasma membrane is a thin, semi-permeable membrane that surrounds the cytoplasm of a cell. It helps the cell by serving as the base for the cytoskeleton and by allowing and preventing certain substances entry into the cell. The cell membrane is primarily composed of lipids and proteins. It is composed of a phospholipid bilayer which has a hydrophilic outside with an inner area that is hydrophobic. The cell membrane also has numerous protein structures running through it. The cell membrane is also a very dynamic membrane, often performing actions such as associating and disassociating with vacuoles in order to help with the importation and deportation of substances from the cell. The cell membrane also has another crucial role as part of the endomembrane system. How secretory and membrane proteins are made using the endomembrane system. One of the central jobs of the endomembrane system is the production of secretory and membrane proteins. To begin this, proteins are first synthesized in the ribosomes on the rough endoplasmic reticulum. After their synthesis, the proteins are then taken by vesicles to the Golgi apparatus. In the Golgi apparatus, the proteins are modified, tagged, and sorted into different vesicles bound for different areas of the cell. Now this is where membrane proteins and secretory proteins truly differ. Through the vesicles, the membrane proteins will be sent all around the cell, where they will perform a host of crucial jobs, ranging all the way from signal transduction to serving as passageways, while secretory proteins will be secreted from the cell by the process of exocytosis.
The nucleus is commonly referred to as the brain of the cell. The nucleus's main function is to store DNA. It is also a site of RNA synthesis. The nucleus is surrounded by a nuclear membrane, which is also referred to as an envelope, and contains a nucleolus and chromatin. The nucleus is surrounded by a nuclear membrane, also known as the envelope. This membrane defines the boundary of the nucleus and consists of an inner and outer lipid bilayer membrane. This membrane contains many nuclear pores which allow molecules to move in and out of the nucleus, essential for communication with the rest of the cell. These pores are protein line channels. The transfer of information encoded by DNA depends on movement of RNA molecules out of the nucleus, and how and when it is expressed depends on movement of proteins into the nucleus. The nucleolus is the largest structure in the nucleus. Its primary function is to assemble ribosomes. The assembly starts with transcription of pre-rRNA by RNA polymerase I. Ribosomal and non-ribosomal proteins and 5S RNA associate with pre-rRNA during and after transcription. The pre-rRNA is then processed into rRNA with help of non-ribosomal proteins and small nucleolar membranes. The rRNA is then combined with ribosomal proteins to form the subunits of ribosomes. These subunits then go to the cytoplasm for final assembly. The nucleolus is also known as the brain of the nucleus and has other functions including sensing cellular stress and modification of tRNAs. Chromatin is a complex of DNA, RNA, and proteins giving chromosomes their structure. It is composed of double-stranded DNA packaged with histone proteins into nucleosomes, forming chromatin fiber, which is also just referred to as chromatin. These are relaxed in regions of the nucleus where transcription is already taking place, going from 30 nanometers to 10 nanometers. The 10 nanometer chromatin is composed of a bead-like repeating unit known as a nucleosome. Each nucleosome is a flattened sphere consisting of about 150 base pairs of DNA wrapped around histone proteins. These proteins are evolutionarily conserved, very similar in sequence from one organism to the next. Lysosomes are specialized vesicles derived from the Golgi apparatus that degrade damaged organelles and damaged and unneeded macromolecules. Lysosomes contain enzymes that break these down. The macromolecules to be degraded are packaged into vesicles by the Golgi apparatus, which then fuse with lysosomes, delivering contents into the lysosome. The Golgi apparatus also delivers special proteins to the lysosomes that become embedded in the lysosome cell membranes. These proteins function as proton pumps, keeping optimal pH inside the lysosome for the activity of the enzymes inside.